Hi, my name is Curtis. I'm a member of the ERIC, a club of student and professional volunteers dedicated to build sounding rockets, rovers and other cool stuff. Today's video features the science payload of our rover ORTHOS. I'd like to mention that the development of a science payload would not have been possible without the support of Laser Components and Biozyme, but more about this later. ORTHOS is one of our main projects. It's a Mars rover demonstrator designed for participation in the European Rover challenge. This event is one of the biggest student robotic competitions in Europe. This challenge takes place in Poland once a year, where up to 25 finalists out of some 88 teams compete in different challenges on an artificial Mars yacht. Within the challenge, different tasks need to be accomplished, including navigation, sample collection and scientific analysis. Our rover is designed in a way to fulfill these tasks. ORTHOS chassis is a rocker-bogie design, which is a common approach for Mars rovers. It enables to keep the mainframe stable whilst driving through rough terrain. Each of its 6 wheels features a brushless DC motor with gearbox to generate enough torque to propel the rover. The front and rear wheels can be rotated for steering. The rover is equipped with a stereo camera on a swivel mount for localization and navigation purposes. ORTHOS is controlled by an NVIDIA Jetson Xavier computer which runs ROS. All high-level functions are implemented on this device. Moreover, it is equipped with multiple electronics dedicated to specific tasks, like power control and distribution, battery monitoring, payload control and so on. These distributed electronics have their own microcontroller, usually programmed in C with an RTOS middleware. To pick up samples and to manipulate the switch panel for the maintenance task, ORTHOS has an articulated robotic arm. The arm can be equipped with different end effectors. Samples from deeper surface layer can be taken by means of a drill. Within a science task, you want to analyze if there's life on Mars, or on the Mars yacht. So we designed a little biochemical experiment or science payload. Our hypothesis is that life on Mars is based on adenosine triphosphate, or called ATP, like every known life form on Earth. So our science payload is designed in a way that we can detect the traces of adenosine triphosphate.
Hello, my name is Tatiana, and today I will tell you about biochemical principle in our science payload. To detect the ATP, we use the principle of bioluminescence. One example of bioluminescence in nature is the well-known firefly. These insects can emit light from their lanterns by using ATP and an enzyme called firefly luciferase. This principle is used in our science payload by means of a so-called luciferase assay. It contains all the necessary agents to create bioluminescence when ATP is present. The generated light can be detected and converted into a measurable signal. But let's take a closer look on how that works.
The assay is based on D-luciferine, magnesium and firefly luciferase. If ATP is present, it forms a complex with the magnesium that shields the negative charges on the ATP. The luciferase catalyzes the adenylation of the D-luciferine, D-luciferyl adenylate, a compound of D-luciferine and adenosine monophosphate. is formed and pyrophosphate is released. The enzyme then deprotonates the adenylate, yielding a negative charge. This enables the adenylate to react with molecular oxygen to form a diacetanon ring. Adenosine monophosphate is released during the oxidation. Diacetanon ring decarbonizes by releasing CO2 and forming oxyluciferine in an excited state. The oxyluciferine tautomerizes between enol and keto form. Only the keto form is displayed here. When the oxyluciferine is returning to the ground state, energy is released in form of a photon with a wavelength around 560 nanometers. The emitted photons can be detected by means of the very sensitive photometer.
To detect the light emitted by the bioluminescence reaction, a sensor is needed that can detect very low levels of light, down to a few photons. This is carried out by means of an avalanche photodiode. These kinds of photodiodes have very high quantum efficiency and they create a very high output signal with respect to the incoming radiation power. But let's have a closer look on how that works. A standard photodiode is a semiconductor consisting of a junction of p and n-doped silicon. At the junction, a depletion zone forms. In the depletion zone, photons can be absorbed, which creates a pair of a hole and an electron, which again diffuse to the anode and cathode respectively, assuming a bias voltage is applied. Thus, a photocurrent is generated. A more sophisticated design is a pin photodiode. In this type of photodiode, an intrinsic layer is put between the N and P dope layers. This yields a greater region for absorbing photons, which results in a higher quantum efficiency and thus in a higher response. Moreover, the junction capacity is reduced, which extends the bandwidth of the device. A much higher sensitivity can be achieved with an avalanche photodiode. The outer layers of the cathode and the anode are high-doped silicon, and between the intrinsic layer and the cathode, a weaker doped P layer is inserted. APDs are usually biased with high negative voltages up to several hundred volts. The layer setup, together with a bias voltage, results in an electric field distribution with a very small slope in the intrinsic region and a high gradient towards the cathode. Like in other photodiodes, the intrinsic region forms the absorption zone, whereas a Bn plus junction at the cathode is the multiplication region of the APD. A photon absorbed in the intrinsic region generates a whole electron pair. The electrons are accelerated in the electric field of the multiplication region, causing further whole electron pairs due to impact ionization. The resulting avalanche of load carriers leads to a strong amplification of the signal in form of a photocurrent. By increasing the bias voltage, the amplification is increased as well. If the bias voltage is raised above the breakdown voltage, the avalanche becomes self-sustaining so that the diode is permanently conducting until the photocurrent is quenched. This enables the detection of single photons. The simplest way to quench the avalanche is the use of a resistor as a passive quenching circuit. The voltage drop of the photocurrent lowers the bias voltage below the breakdown voltage. The voltage drop in form of a pulse can be analyzed by the readout circuit. In our design, we use the SAP500T8 avalanche photodiode. We are happy that Laser Components generously sponsored us such a high-performance device. The SAP500 has excellent quantum efficiency, high gain, and low dark count rate. Its key performance parameters outperform those of APDs from other manufacturers. It can be either used in linear or photocount mode, whereas we use the latter one. The T8 version, which Laser Components gave us, is equipped with a two-stage thermoelectric cooler. Cooling reduces the dark count rate and therefore increases the signal-to-noise ratio. The device is equipped with a temperature sensor, which enables the control of the temperature. As the breakdown voltage is temperature dependent, the constant control temperature is preferable. So far for the basic principle of our scientific approach. Now we want to have a look on how we have put this principle into practice. The system features a revolving drum which can be charged with 6 disposable sample tubes containing a chemical that can break up cells. By rotating of the drum, each tube can be positioned at different interfaces, for example at the loading port or beneath the photometer. The sample tubes are enclosed in a light-sealed casing to block off ambient light from the photometer. A servo motor attached to the revolver can rotate the drum. To add luciferase assay to the sample, a syringe unit driven by a stepper motor linear axis is used. The APD is mounted on a PCB that features a readout circuit for the element's temperature and the photon count signal as well. The APD unit is mounted on top of the science payload. It is housed in a casing so that no ambient light can hit the detector. To separate a small sample for the analysis, a feeder system is incorporated. A funnel and a sieve deck separate stones and other materials that are too big for the sample tube. The feeder is equipped with a rocker that can be set to different angles. To separate a small amount of the material, the rocker is swiveled just below the edge of the feeder's ram. This allows some small portion of material to get stuck at the edge while the rest of the material is disposed. The rocker then is swiveled open so that the sample can fall through a small funnel into one of the sample tubes positioned below the feeder system. After the sample is loaded to the tube, the rocker swivels up again so that no light can enter the payload. When the sample is loaded, the sample tube is rotated by the revolving drum to the luciferase feeder. Luciferase then is added to the sample. The sample tubes can be driven on a vibration plate. The plate can shake the tubes to mix the sample with the chemicals necessary for the ATP detection. When the chemicals are mixed, the sample tube is positioned under the avalanche photodiode. The lens focuses the emitted photons onto the sensor. Now let's see how the science payload is set up operational.
BioZyme generously provides us with all the equipment for detecting ATP. First of all, we have this ATP biomass kit. It includes the extractant solution, which breaks up the cells in the sample and set the ATP free. It includes also dialant B and ATP reagent. Mix them together yields luciferase assay, which reacts with the ATP and emits photons if ATP is present. In the set we have also ATP standard, which we use for calibrating of our analysis unit. To charge the analysis unit with the agents, the BioZyme provides us with such adjustable pipette and the set of single-use pipette tips. And the last but not least, we got a bunch of sample tubes which we use in our science payload.
To load the sample tubes into the science payload, the maintenance hatch has to be opened. The sample tubes are filled with the extractant so that cells in the sample can be broken up to set the ATP free. Up to 6 sample tubes can be charged to the payload. One of the tubes gets a specific amount of ATP standard solution in addition to the extractant. When all tubes are loaded, the maintenance port is sealed again so that no ambient light can enter the casing. In the next step, the syringe is filled with luciferase assay. It contains enough assay for 6 samples. The syringe is mounted to the linear stepper axis. The readout circuit then needs to be connected to the science payload control electronics. The payload is now operational. The science payload can be controlled by a graphical user interface. Commands and data are exchanged over a CAN bus. To increase the signal-to-noise ratio, the avalanche photodiode can be cooled. The set point of the temperature controller is usually set to -20 degrees Celsius. The actual temperature of the APD is displayed in the user interface. Let's activate the avalanche photodiode by increasing the bias voltage over the breakdown voltage. For this device, a voltage of -115 volts is suitable at a temperature of -20 degrees Celsius. We see that we get a photon count signal. The signal is around 250 counts per second. This is our noise from which the signal must stand out. 250 counts per second is quite low, which underlines the excellent performance of Laser Components SAP500T8. Let's deactivate the APD again. Now we drive sample tube number 1 to the loading port as the sample feeder. The sample is now filled into the funnel of the feeder. The rocker is opened so that a portion of the sample can fall into the sample tube. The rocker is toggled a few times close and open so that all of the sample is fed to the payload. Sample tube number 1 is now positioned under the syringe unit. 400 microliters of luciferase assay are added to the sample. The sample tube is now driven to the mixer. The shaker is activated to mix all the contents. The sample tube is now driven beneath the avalanche photodiode. The APD is activated. A strong signal of around 8000 counts per second can be observed, which stands out sharply from the noise. Now we repeat the same sequence with sample tube number 2, except that we do not put any sample into the tube. This is our blank sample. When we activate the APD, we observe a photon count signal in the order of magnitude of the background noise. That means that we do not detect any bioluminescence reaction at all. This indicates that our system has not been contaminated with ATP during the setup. We have proven that our sample contains adenosine triphosphate, which means that there's a high probability that it contains traces of life. Sample tube number 3 contains a specific amount of ATP reference solution. We analyze it in the same way as the former tube, again with no sample added. Probing the reference sample yields a signal of around 500 photons per second, which is about twice as the background noise alone. Comparing the peak signals of the sample and the reference and correcting by the background noise offset enables to assess the amount of ATP contained in the sample.
So, today you learned how our science payload is designed and operated.
I'd like to thank again Laser Components and Biozyme for their support. We are happy that we got hands on such cool products and that we could use them in our design.
If you enjoyed this video, please subscribe to our channel and leave a comment if you like.
Bye!